big camera, so the big, we use a 5-gallon bucket for a lens cover, which is amazing. So it's got some just dynamite optics in it. And that camera housing's been around for a while before there was even an HD camera to put in it. It had what we called a 3-chip camera. The old Sony monitors that go with it. The old tube tube monitors, you know, the
CRT.
CRT, yeah, that's what I'm after. In a 19-inch rack. So the titanium housing and the lens, custom ground lens with a custom diopter in it for the optics is just beautiful having one of those to look at the ROV with. And the other big advantage is it having the fixed length tether between Argus and Hercules. It just frees up Hercules. So I mean, this whole watch, I've barely been touching the controls. It's just kind of floating along in the current, like one of the little eel pouts almost. You know, it's completely free. So you don't get that feeling with a single body system, or even a caged system that has a tether that can go in and out, because the fixed length tether has the floats, the 2 floats on it, and then the weight. So it's basically neutrally buoyant behind Hercules. So it's really quite enjoyable to operate this vehicle, because it's, you know, it's almost like it's floating free all the time. You don't have the forces of the tether tugging on the vehicle, which on most other vehicles I've operated, you really feel you really feel that leash pulling on you. So if I had to,
but I guess it requires good communication with the ship, with the pilots and the navigators to keep this sort of delicate balance.
It does, yeah.
In place as
navigator.
Conditions. Yeah.
Navigator really has to be one step ahead of the pilot the whole time and planning
Predicting
the ship
future.
moves.
Yeah, it's well in the deeper water, it's kind of that pendulum you can think about. You know, like moving the ship around on the surface. It takes a while for Argus to actually move, because the cables will kind of you know, it has a lot of depth to go through before Argus feels that move. So the deeper we are, this is 800 meters, and it's actually pretty, it's not so bad. You know, we call in a move and we kind of know, you know, Argus will move shortly after in the direction we want it to go. But we were, there's other considerations when we're 3,000 or more meters deep, because then kind of have to, there's a lot of layback. Will be, you know, the vehicles will actually be 200 meters behind the ship. And so then you can't just stop on a dime and go sample. You have to backtrack the whole operation and all that.
There's some pure magic happening in this van with the 5 people that are in here to make all this happen. To, not only the navigator, but the Argus pilot keeping the perfect delta, as we call it. So he's always 15 to 20 meters above Hercules as we go up and down through all these valleys and cliffs, and he's adjusting the heading as well as operating all the ancillary equipment on Hercules. And then the video is always magically perfect auto iris happening, even though it's not auto. You can tell when that doesn't happen, but it's always, it's just about always perfect. And then everything is somehow magically time correlated and recorded in the back row back there, so we can later go back and find that perfect shot. So
yeah, when you go and you look at all the event log notes, it's just like every moment's recorded, and you realize that there's a human back there typing that all in.
Yeah. Without Taylor Ann doing that diligently, it would just be a photograph somewhere in the middle of the ocean with a timestamp on it.
Yeah.
So we have Guy Cochran asking if we can put nav back on channel 3. Is that possible?
Sure.
But I understand why it's on where you did, because so that people could see.
Yeah, it was just
This was a great portrayal of what's going on.
it was
of
showing
what you were just
the
all discussing. Yes.
ROV nav. Ready?
Oh, nice. Might have to mosey on upslope.
Yeah.
Unless you want me to go down
Yeah,
a bit.
sorry. I'm mindlessly coming down the hill there while droning on.
No, that's fine.
Forward now.
Thank
Giant
you for
boot
explaining
sponge
that.
line.
I thought it was good explanation.
I'll zoom out a couple
Of,
clicks for Guy on shore to take a look at where we're at. So we're right near 3C, and we're just kind of going north-ish up these, following these contours up until we want to depart and go back, or waiting other instruction.
Yeah, we're just going to check this out for a little bit and then probably need to get to the other side of the fault.
So that whole horseshoe shape is the fault scarp itself on the dive plan?
Let me see what's the horseshoe shape. I'm going to open up the dive plan.
The kind of the valley we're going to cross over to get over to 3D that goes north.
So the way I understand it is points 3A, B, C, are all on one side of the fault.
Okay.
And I think D, E, maybe, I'm not sure if D belongs also on the other side, but maybe Guy can say, but definitely we want to get to the other side of the fault. Pretty sure D is on the other side.
Okay, that makes sense.
But I personally have a hard time reading these maps. I have a hard time figuring out what is going down, what is going up. I do not have a knack for that. And when I've gone hiking and gone off trail, if I didn't have other people with me, I would have gotten lost. So.
Yeah, Guy is saying in the chat, he doesn't know why 3D looks so deep.
3D looks deep. Yeah, it's downslope about 100 meters. Well, it's horizontally about 1 kilometer away, but it's downslope about 100 meters, I think, ish.
So is 3D on the other side of the fault?
Looks like it is, if I'm reading that correctly.
Yeah, I think so too. And then, Guy, while you're listening, so are D, E, F, and G on the other side of the fault?
He says we are on the fault now.
Yes. But when we there's a canyon that cuts through the fault.
And he also clarified, not looks deep, that 3D is deep. And
The fault is on both sides. I get that, but I guess I'm trying to say, is D on the other side of the fault line? So, because one of them as it moved, lifted up, and the other one did not. The other side of the fault line. No, it's way out in the canyon axis. Okay. I need to take a geology class.
You'll have to take more than one. It's
Well,
not
then
that
maybe I'll
easy.
just stop. I'll quit. I'll quit where I am.
I've failed
He's,
geology like at least half a dozen times.
so Guy Cochran is asking to zoom out.
Zoom out on HiPAC, Roger. That has all of our points in view, I think, or it's got the rest of them. So we're right around 3C. Then there's 3D, F and G.
But did you meant zoom out on the nav? Yeah, that's what they did.
I've seen one of these guys
Thank
up
you.
close. Landing.
Oh yeah, nice Paristonella.
Go
Yeah,
ahead,
can we
Steve.
look at it up close? Thanks, Steve.
That kind of pink color it's got is from all of the brittle stars that are all over it. This is very
yeah.
typical of the species.
Is the brittle stars, why do they like Pyrostenola so much? They're not feeding on it. They're just using it as a place to put their arms
Just
out into the
to
current,
get up in
correct?
the water column. Yeah,
Yeah.
I don't think they really know why they particularly like this species so much, but this is very typical that you'll see Paristonella just absolutely covered in brittle stars. It might be that maybe they can hang on to it more easily. But yeah.
Yeah, you
They're
think
just
about how
using it to get
different
up
it
in the
is
water.
from the Paragorgia when there's no brittle stars on it. And that would still be up in the water column. Gotta be something.
Okay, so Guy is recommending that we alter the dive plan or the transect, rather than
Okay, Steve.
flying from here from 3C to 3D, to stay on this side of the fault and to cross over, to just move north, stay on this steepness, and then move over to 3F through a water.
I want to stop him for a minute.
Yeah, Roger that. Bridge nav.
So we would
Let's hold
bypass 3D and 3E. Does
position.
that make sense?
Yeah, it makes sense. 3D was pretty deep, and then E was like a continuation. I was looking at it in 3D, kind of a continuation here. So we're going to go essentially up to, I'll just drop a rough target, so somewhere right around there.
Yeah.
And then we will go directly across to F or something thereabouts.
Yes.
Okay.
Perfect.
Can do.
Thank you.
Yeah, and I think our goal is to stay in the steep stuff as much as possible as we kind of transit up to that target.
Sure. Yeah, I'll pass that along too when we. I think that'll be the next watch once it starts to. There's a bit of a flat bit there. I'll tell them to try to avoid that.
Stay away from the mud.
Yeah.
So here's a dead heterocony, and you can see that's a sponge, the one that we looked at just earlier with the beautiful orange frilly collar. This is the same species, and it's dead, and you can see that it's the
quick zoom there, Steve.
Framework can form sort of another habitat.
Yep. It's got other sponges already growing on the outside of it. So there's a live Pharea, a couple of clumps there. And so since this is one of the ones with the fused skeleton, this is basically a giant, very elaborate rock at this point, and it's now habitat that other corals and sponges and hydroids can grow on.
Oh, and there's an, is that an umbellula or an umbellella? What is that?
That is a Giovannia cup coral.
The one on the left.
On the left, yeah, just going off the screen.
Yes, okay.
On the left?
Right.
Oh, there he is. Yeah, I see him.
Wow.
But you knew which one I was talking about, right, Meredith? The
Yep.
But those are more on a stalk.
That's on a stalk, and it's actually attached to a rock right there.
Okay.
Zoom in there, Steve. It's a cup coral.
Posing.
Strange place for a cup
I thought
coral.
that was this. Yeah, I thought that was a stalk right behind it.
The guy is recommending that we go back upslope a bit from here so we get back into the steep before we proceed kind of north-northeast.
Sure.
Yeah.
Can do. See if there's anything, I'll just keep Argus here. We'll see if there's anything steeper toward upslope at the end.
Yeah, looks like there is.
Yeah.
We like going uphill. It's easy.
So I'll give you a 10-meter bump up or something like that, maybe 15 meters.
So is this an Isadella, the one with the...
You can look 045 with Argus for a while.
Yep, that's Isadella. You can just see the long tentacles.
polyps? Yeah, long tentacles, yes, not polyps.
I think they are long, like elaborate polyp body.
And
Great
is there a
snap.
reason for them to be so long at the base there? Is it like an anti-predatory?
I don't think anyone knows. Just a quirk of evolution.
See if we can get to that. I don't know.
There's a big boulder.
A lot of sponges on it. One little Swiftia coral.
Well, this is, I think, a great course correction, so we can continue in this wonderful habitat and we don't have to fly. Somebody else gets it on their watch.
Yeah, the next shift gets it.
Yeah, or maybe the one after that.
Crab.
Nice little thorny heads.
Come on, ROV. I'll take you on.
Yeah.
I got bigger
I was like-
manipulators than you. Cracks
Like
me
when
up.
they're so feisty.
There's a moon coral, I think. Or moon, what would we call it? Moon sponge, I mean.
Moon sponge?
Is that on the left, the green tennis ball looking one?
Oh, yeah. Yeah.
Oh, we were gonna look at it. Can we look at it?
Yeah.
Sure. Let me see if I can.
Who is speaking? Was that you, Steve, or was that Rennie?
That was Steve.
Okay. Oh,
Yeah, usually I
something
don't.
spooked it.
Usually I don't get IDs like that, but I was excited.
No, I'm just trying to put the voice with the person.
Sure. Oh yeah.
Oh, sorry. Okay.
Now I see. Yes.
There's the moon.
Yep.
Very moon-like.
This one's Just a little guy. When they get bigger, the craters will get like big and shallow and really elaborate.
So does it have a genus name or?
This is a Ladhuncunia.
Now, how do you spell that?
I just
I'll
spelled
look
it in the
in,
science chat for you.
Okay, thank you. You knew I was going to ask.
Yep.
Thank you.
And we've got a little Swiftia coral right next to it.
The one on the left there.
Yep. That's the we've been seeing two varieties of those sticks. One is Swiftia simplex, and that one has the darker red polyps. And this one is Chromoplexara marquis that has the white polyps.
Pretty cool.
Mm-hmm.
We could move on.
Roger, moving on.
Thank you.
Nice find, Steve.
Looks like some clavularia there because it's so sparkly.
Yep, that's more clavularia. I like how it all looks like a big fluffy white poof.
Clavularia or the sponge here?
The clavularia.
Yeah, I agree with you. They're just look festive. There's a big barrel sponge up ahead.
Yep. I'm trying to see what that white thing laying over is. I think it might be a bamboo skeleton just laying on the bottom there. Yeah, that's a bamboo skeleton.
So what's the name of these barrel sponges that have sort of brown on the outside and they're completely white on the inside?
So normally we would call that Rhabdocalyptus.
Mm-hmm.
Seen a
But
yellow
they're,
barrel sponge before. Yeah.
Yeah. So this could be possibly Rhabdocalyptus or Staroclyptus, but a yellow morph. It could be a Staroclyptus that's starting to not do so hot, although I'd expect it to be fuzzy or starting to look brown on the inside too.
Oh.
What we found when we sample some of them that are this yellow color is once you get them into alcohol, the yellow color will leach out.
So these are again like hydroids living on the outside.
No, so that's actually these sponges have kind of this cloud of spicules around the outside edge, and in these species it gets covered in kind of dirt and stuff. So that's actually sponge spicules.
Oh, thank you for
Yeah, you
explaining
can
that.
you can see a nice
So
view
it's
of them right
just
here
from,
on the lip.
yeah. So the spicules just trap, it's like a shag carpet.
Yep.
So they're trapping any of the detritus and sediment that wafts by in the current. But wouldn't that clog their the pores that they need that are filtering the bacteria through? And
It doesn't seem to in these species. Yeah, it's like just this loose outside matrix. And on some of the Rhabdoclyptus, it can be just like this thick layer of dirt, but then they're just perfectly white on the inside, and it doesn't seem to affect their ability to filter feed. And if you look at this sponge under a microscope with these spicules, they actually have part that's embedded in the sponge wall, and they form these umbrellas. And what you're seeing sticking out is basically the handle of the umbrella.
so the flow of water for these sponges is from the outside in, correct?
Yes.
Just
Yes.
yeah. And then because I'm remembering when I was doing research in the Caribbean, and we dive on coral reefs with some sponges, and we bring bottles of dye with us, and we squirt some on the outside, and you could then see it come bubbling out like a volcano.
I've actually always wanted to do that with some of these deep sea sponges. Bring down a little bottle of dye that Hercules could squeeze and
Yeah,
see
we
how those
should
current
make that
flows through.
happen.
That'd be fun.
We've
We
done
should
that.
make that. We should
We
try
just
and
need
make that happen.
I just need a rubber bag full of fluorescein and a little nozzle. Waft it around the sponge with a manipulator and watch it respirate.
So have you done it before, Dan? Is that what you were saying?
Yeah. Many
wow.
times.
Well, Meredith would like to see it happen. Can we see it happen? She's going to be on this shift a couple times this week.
Talk to Sally at U of H. She's got it down to a science.
I think it'll depend on
So
if they have fluorescein
where is
on board.
Sally?
University of
Seriously,
Victoria.
Dan, where?
What's that? She's one of the scientists at University of Victoria. She does studies of sponges there on Vancouver Island. It's the big inlet off the strait there. Oh God, it's been years.
So do you have any fluorescein on the ship now?
I don't know if they have any fluorescein in the lab.
I don't think so. Yeah.
Taylor Ann's shaking her head no.
Yeah, I don't remember that being a standard.
It would make for a very good educational demonstration.
You can look at the chemical inventory, but I don't think, unless a scientist had left that on, and I don't recall ever having done it.
I basically take a soft bag of it down, and it just gets enough pressure to waft out of basically a syringe needle.
Can we get some food coloring from the kitchen?
Yeah. Kool-Aid mix or something.
Yeah.
It's another Ladincunia.
Is that crab moving something, Or does it just have a,
I think he's got his claws in
I
a
think it's
weird. He's just
yeah.
up and ready to go.
Another baby fish there.
okay.
Yep, that's another baby thorny head.
You guys know I'm still milking this uphill till shift changed.
Yeah. Well, I don't really see too much. It gets so much steeper, and then it'll get quite flat again.
So yeah, still got a wall of red in front of us.
Yeah.
Yeah, our scientists who are going to be the science team for the next shift are starting to join us in the science chat.
Welcome.
Erica
Yeah.
Frew has just logged in.
Erica Frew and Liz Clark are coming on together. This has been a pretty cool watch shift,
Yeah,
I have to say.
the highlight was
It's very nice.
definitely the big pair
Bridge
of
nav.
gorges.
Yes.
Yeah, do we have
10
those
meters,
posted
045.
online yet?
Thank you.
Those posted already, Taylor Ann, or they gotta wait for the outreach people to wake up?
Yeah, the pictures, yeah, probably have to wait for. I marked them as a highlight though, so.
Good. So Meredith, do you want to stay on and explain to Erica and Liz how we've changed the transect? And then Rainey,
Sure.
I assume you're going to tell the navigator who's coming on what the plan is?
Yeah, I'll pass all that on, and then if they need to go over it, he can zoom out and talk it through.
Right, good.
Just dropped a rough target, so it's TBD on.
Yeah. Yeah, and I guess we just need to explain to the next science team how we're staying sort of on this steep slope area and sometimes popping up over, but if you can just find that the lip
Yeah.
just moving up and down, and that's where we find it. Find the good area of the corals and sponges.
Yeah, it was just
the
below
lip.
the lip.
Yeah.
Yeah. And I'll wait. I'm going to stay on, but I'll wait for Liz to show up and then.
Yeah.
Bridge nav.
Nice pom-pom anemones there.
Add another 10 meters. Thank you. It's 20 towards you.
So if I wanted to review what we just saw, I've been hearing people say, "Oh, I'm going to watch it on YouTube." Is this it's all on YouTube already?
Yeah.
Yep.
You can scroll back.
And I would go. And what would I search for on YouTube? What?
I think if you go to our YouTube channel,
Yeah,
Nautilus.
there's a playlist of videos, I think.
Yeah,
And they're sorted by, like.
And the current dive, I think, should be, the most recent. Then you can rewind it, I think.
Yep, that's right.
Yeah,
Okay.
so the.
But you have to have already watched it to rewind it.
Yeah, so the current dive, you can go back up to 12 hours through either nautiluslive.org, or if you're on YouTube, there's a Nautilus Live channel that will be broadcasting this dive live, and you can rewind up to 12 hours. And then in about 24 to 48 hours, this whole dive will be posted as a dive you can watch. But that's usually a full day or two later.
Thank you. I was wondering, I'd hear people say, "Oh, I reviewed," and I thought, how are they doing that? What are they doing? Thanks.
Oh, no worries. Yeah, often like if I'm starting a shift and a dive has been going on for a while, in addition to getting the review from the previous watch team, I'll quickly just do kind of a fast forward zoom through the last 4 hours, visual picture of what they've sampled and that kind of thing.
Oh, that's pretty smart. Just go
Yeah.
zip through it.
Yeah, and that just
Those
makes
are
sure that
sweet.
like I've
Fast
seen,
forward.
Yeah.
when someone says we've sampled a plate sponge, that way I can see like, okay, it's this type of plate sponge versus another and have just a visual image in my head of what's already been sampled during a dive.
Oh, nice. Yeah. Guess that's
And
the,
what are these two corals that look like two little hills?
On the left?
I mean sponges, sorry.
Yeah.
They're white right here on the rock.
That is another like kind of unknown sponge, and I think this is one of the ones that Abby sampled on the shift just prior to ours.
Okay, so we don't need to. I'm gonna look at.
Yeah, we've been seeing this one
log.
throughout the dive, and I'm pretty sure they sampled it.
Okay. This has been an exciting watch. So Liz and Erica, while you're listening, we found a ledge. Meredith, why don't you tell, because you can speak about the species more. Give them a highlight of what we saw.
We've been kind of moving up and down through the steep section of this wall we've been on, moving up to the top and then moving back down to the flats, and they're both flat with a steep wall. And just over the lip of the steep wall at about 2 o'clock, we found some ledges that had just probably meter wide, big Paragorgia fans that were quite striking, and also probably 5 or 6 other species of coral, Peristonella, 2 species of Swiftia. There were 2 species of Paragorgia, Gersemia, and mushroom corals all in that area. Oh, and the Isabella bamboo corals. So in moving up and down this steep wall feature, the area just over the top lip seems to have the highest density and diversity of corals. And Erica says, have coffee, I'm ready.
Well, then I will jump off. And Meredith, it was a pleasure. And Dan, great driving on Herc, Jake, you two on Argus, Rennie, thanks for the nav, and Steve for the video, and Taylor Ann for your great data logging. And we'll see you again on the midnight shift, I believe, because the next 12 to 4, this dive ends at 12, so the 12 midday. So we'll be on deck. But thank you.
Good. Yeah, thank you.
Thank you, Lisa.
Thank you.
Thank you.
Good night. Have a good
See
rest.
you tomorrow,
You too.
Lisa.
Okay. Bye. Thank you, Meredith. Bye.
Bye. And Liz and Erica, I'm staying on to go over just the nav stuff with you whenever you're ready to jump on.
Good morning, Meredith.
Good morning, Erica.
What's the word?
Word is we have lots of these lovely bamboo corals. And I'm going to jump over to the nav screen real fast so I can better describe what's going on channel 3. We've basically decided, under the advice of Guy, who's been on through the dive, to skip 0.3D entirely because it's just so deep. I'm going to quickly jump back in the notes and check one thing. Yeah, we're going to skip 3D.
Let me write that down.
Skipping 3D and skipping 3E.
Okay.
So if you look at the nav screen, you'll see a target just northeast of us that says 103008.
Copy? Yep.
Yep. And so what we're doing right now is just trying to stay in the steepest portion of the wall and just working our way up to that. And then the goal is to then jump straight across to 3F and then work up that steep side, other side of the canyon. And that was the plan that Guy's been recommending. And yeah, the most productive area for coral and sponges has been just over the top, just like on the steep side, just off the lip of the top edge of this steep section.
Okay.
And I think we haven't done a transect in a while. We've just been looking at some stuff and taking a couple samples. And so they should be ready for another transect soon, if you want to start with that. And then we have recorded all the samples so far in the data recovery report. So we're currently on.
Great.
We're going to do a watch change in video chair. Nice watch, everyone. Thanks.
Thanks, Steve.
Thanks, Steve.
Yeah, you guys have been busy. I'm looking at the collections.
Say most of that was Abby.
Of course it was.
Can you hear me, Erica?
We've got
I can hear you. So, Liz, I've given Erica our whole plan and status. So unless you need another update, I will jump off and let you guys take over.
Thanks a bunch, Meredith.
Yeah, thanks. And I look forward to following along as I get ready to go to the center this morning.
Oh, goodness. When do you sleep?
This afternoon. I'll sleep this afternoon.
Okay, good.
All right. See you later.
Have a good one.
You too.
Morning, Erica.
Hello.
Boy, these 4 o'clock watches. The scramble to get all my IT working. I heard that we're just headed along this wall and occasionally tipping over the ledge to peer over. What depth we're at here? Just looking. We're doing a watch change now, right?
862 meters.
Yeah. What's interesting about this area right now is this is what I expected the area that we were on the first Santa Lucia dive to look like. And I think it's what the geologists thought it would look like too. These plates, this sort of this sedimentary rock that is broken up. Looks like it's been thumped from below, sort of like an asphalt driveway that's breaking up. But we saw completely different rocks, things that looked like they'd tumbled from the top. But this looks like kind of like the area that we would see in some shallower water too. So it looks more familiar to me than our last Santa Lucia dive. Sure is pretty though. We're just waiting to do a watch change, right, Erica?
Yes. They've seen lots of different corals and sponges. I've been watching for the last 15 minutes or so, and the sponges are really different. I hope Melanie wakes up and logs in and tells us what some of these are.
Yeah, and I think Abby was up for part of the time, getting some samples.
That's what it sounds like, yes.
I wonder if we have what sort of sampling space we've got left.
1, 2, 3, 4, 5, 6. Looks like 9 samples so far.
So while the pilots are doing a watch change, I'll get some people who might be listening in to us oriented. If you go to the quad view that's on the Nautilus Live screen, that large scale map tells you where we are, which is off the Morro Bay area in an area called Santa Lucia Bank. And then if you go to the left to the other map, what you'll see there is, I hope I have to get the quad view of myself here. Yes, you're seeing a detailed map, more detailed map with depth contours on it. And we are trying to see where we are. I think Nav's going to have to tell me where we are on the waypoint.
We just did our watch change.
Okay.
So I can zoom in for you. My understanding of the plan is we're going to continue along this kind of steep portion, kind of following this contour up north.
Mm-hmm.
And then we're going to head west down slope. Probably you guys want us to quickly traverse this flat portion and then go to 3F. Does that sound like the plan you got?
Yes.
Great.
That is what I heard also, Ben. So
Awesome.
we're on the same page.
Okay.
It's always good.
So for the people at home,
right?
We're talking about those dots on the map and going which waypoint to go to and what's the cruise track we're going to take with the ROV. And that's what we're going to do. Except I got to get my glasses. So to kind of read the dots. But
Meredith also mentioned that it's been a while since they've done a transect, so we might want to think about
that.
Roger, it's been, we're 130 meters away, so we're good to start that.
What do you think, Erica? Should we do a transect right now?
As soon as the pilots are squared away, I think, if it's okay with them. Yeah.
Roger.
Yeah, sure.
Yeah, I think we're going to do 3, 4, 5.
3, 4, 5 doing on the contour?
Yeah, the contours seem, Brenny says they've been pretty accurate, so we can just trust that.
3, 4, 5.
And
sounds like Sesame Street.
Yes. Count Dracula.
Count Mapula.
Mapula, yes.
Oh, Lord. Right.
It is 4 a.m.
Yeah, just so whenever.
Yeah, I'll just get myself squared away.
Yeah. Let's see what that looks like in the ESO.
Maybe 3, 4, 5?
3, 4, 5, Roger.
This is 3, 4, 5 right now.
I'm going to reset you real quick.
Sure. Oh yeah, there is a good current. I'll get in front of Argus, then I'm gonna be ready.
Let's see.
Got you surrounded.
Roger.
All right, not to be gratuitous, but I might do 347.
Roger
A
that.
non-5 move, but I think that will be a little better. I don't want to have to change heading as we do that.
So when we say we're going to do a transect for people listening in, it's we're trying to quantify how many corals and sponges and fish and all the things that we see. So we do transects, and that is that we have the ROV go along at a pretty fixed speed and height above the bottom for a distance of about 200 meters. And that's what we use that to count all the organisms. So we can compare different sites. So we use that same height above bottom, speed over bottom, and distance. And we do that all over the sites that we're surveying. So we won't stop and peek at things while we're doing a transect. We'll just click along.
All right, Ben, I think I'm going to just crab along a little bit so we get better visual contact.
Roger.
Science, is this a good view for you guys? We can keep a little ahead so we can see what's ahead of us, but with the slope, is that all right for you?
Yeah. I think it's okay. Erica, what do you think?
Yeah, I think it's fine. They can count what's in the field of view. They always ask us to get closer, so this is good.
But in this case, you're going to bang into that big boulder up front there.
Not Jess.
Jess is too good for that. I've already had my tea for the morning. I'm good. Thank you.
Nice.
All right, Ben.
So your heading is 020, and mine, I'll move you along 34. I'll just do 345.
345 is great.
Roger.
What happened to 347?
I gave up on my ambitions. Too ridiculous. All right.
Everybody's doing their own thing this morning.
Okay, so we're ready to start, are we?
Roger.
Yes, ma'am.
Yeah, let's do it.
All right, drop to target.
Are lasers on, Jess? Or
Yep.
am I just missing them? Okay. I'm just missing them.
Have they been seeing it looks like a huge round boulder in front of us. Have they been seeing a lot of those rounded boulders? Anybody know?
I have not. I don't know.
I'm going to check the science chat, see if anybody answers me. So for the people at home, Erica and I are sitting I'm in my dining room. Erica, where are you? In your living room?
No, I'm in my mission control at home office.
Okay. My office wasn't big enough to hold the monitors and the computers. So, and Lisa Winnick, who just got off watch and is one of the co-leads for this cruise, Nautilus 123 actually is the cruise, says that she's seen some of those large boulders and the large Paragorgia that they saw, which was a coral, was on those boulders.
Data, can you tell me what time this transect started?
We haven't started the transect yet.
Yeah, we're
Oh,
just
okay.
We're
waiting for
just
the ship
setting up.
move.
Sorry.
But
No
we
worries.
technically will start right about now. Okay, go ahead and log that if you want, Mary.
All right.
And Mary, do you know what transect number this is?
This is transect 5.
Roger, thanks.
Okay. And we're off and running on our transect. It's a pretty interesting area. People who were on the watch before us were telling us that saw lots of sponges that they'd never seen before. One sponge was kind of two mounds, I think.
Oh.
There's a nice barrel sponge in front of us with the crab sitting on it.
I think I got a little overzealous there. Not off to the races yet.
Do you feel the current in this area when you were moving? Yeah.
There was a good clip and I took off, but seems it's only on the tops. All right.
And I called in 0.3 knots. Few.
Roger.
And that redfish is a thorny head, and I'm going to say it's a short spine. Erica, tell me if I'm right or wrong.
I would probably go long spine at this depth.
See? And what we're seeing are sponges, these white, sort of more fluffy things, and then these branching things are corals, deep sea corals. Most people have heard of corals from coral reefs, but these are corals not completely unlike coral reef corals, but they don't have the advantage of being in the sunlight, so they don't have any symbiotic algae, which almost all tropical corals have. So these live in the dark, but they feed in much the same way, and in some cases look a lot like tropical corals. Probably not as bright in the coloring in a lot of the tropical corals. Erica, I'm going to go on mute for a second while I get my coffee off the coffee maker.
Okay, we're seeing a couple different types of coral. Go ahead.
Sorry, just debating about it.
Yeah.
Maybe one ship move that way, because then I can get Argus a little off the wall there. Since
Yeah.
and then then I'll kind of lateral across its screen a bit.
Well, 345 will keep us off the wall. That's what you're.
Sorry? 345 will keep us off the wall?
Yeah.
Okay.
And 350 will bring us closer.
Oh, you're right. Yeah, then 345 stays good.
Roger.
I'm going to stay on the little bit shallower end of the screen.
Nice. So do we know if they collected any of those rare unknown sponges?
I know that our sponge guru was on.